Hello everybody, this is the second part of my auditory lecture. So in the first part I talked about the properties of sound, which is the stimulus for auditory system. I also talked about the anatomy of the ear and different parts of the ear and the structures and functions. And I ended by talking about the cochlea. So this is a cochlea. You know that cochlea is a coiled snail-like hard bony structure filled with fluid, and inside the cochlea you have three chambers or cavities separated by two membranes. So let me just quickly review a couple of things or add a couple of things. So if you just make a slice cross section of the cochlea, you will see inside two membranes, right? Vestibular membrane, the upper one, and basilar membrane, the lower one. Okay, and three cavities. This is the scala vestibule filled with perilymph. This is scala media filled with endolymph, which is rich with potassium, and this is the scala tympani, which is also filled with perilymph. Okay, and this is the basilar membrane, and on the basilar membrane you have the organ of Corti, right? And inside the organ of Corti you have two types of hair cells, inner hair cells and the outer hair cells. And if you see the hair cells, so this is an inner hair cell. The number is much less than the outer hair cell, and these are cilia, right? So inner hair cell, and these are the outer hair cells. Okay, nucleus inside and cilia, stereocilia, and the largest stereocilia is called the kinocilia, right? So this is inner hair cell and outer hair cell. Okay, and I explained the mechanism, how the cilia bend and ion channels get opened, potassium gets in, as well as calcium, and the neurotransmitter glutamate is released, right? Now you see from the hair cells or sound receptor cells, the afferent fibers go to the cochlear spiral ganglion. I talked about this in last lecture, cochlear spiral ganglion. And inside the ganglion you have two types of neurons. One type of neurons are called type 1 bipolar. So bipolar, these are also called type 1 neurons. Okay, and another type of neurons are pseudo unipolar. Okay, and these are type 2. So pseudo unipolar type 2. Makes sense? Okay, so remember, type 1 bipolar neurons receive the signal from the inner hair cells, and each hair cell sends signal through a single afferent fiber. So you see, this is the inner hair cell, and from each inner hair cell a single afferent fiber takes the signal to the bipolar neuron. So each bipolar neuron receives signal from each individual inner hair cell. Make sense? And the pseudounipolar type 2 ganglion cells, each ganglion cell receives signal from multiple outer hair cells. So you see here, from multiple outer hair cells, the signal is taken by one fiber to one neuron. Make sense? So that is the difference. And as I told you, that 95%, about 95% neurons in the spiral ganglion are receiving signal from the inner hair cells. That means the signal from the ganglion cells going to the central nervous system, most of the fibers are taking signal from the inner hair cells. Make sense? So the sound signal is mostly carried by or transmitted by the bipolar type 1 cells to the cochlear nucleus in the brain. Okay, so this is the cochlear first nucleus that receives signal from the auditory nerve, cochlear branch of the auditory nerve. So cochlear nucleus, and these fibers are the cochlear nerve fibers. Auditory nerve has two types of fibers, cochlear and vestibular. Since these fibers are taking sound signal, carrying sound signal from the cochlea, that's why these are cochlear nerve fibers and take the signal to the cochlear nucleus. Okay, so that's how the signal gets out from the cochlea. Okay, or taken out from the cochlea. Now there are two other very important structures in the inner ear. One is the vestibule, okay, which is responsible for linear acceleration. So vestibule picks the signal of linear acceleration of the body or head, and the vestibule is also a bony hard structure filled with perilymph. Okay, and if you see here, vestibule, if you see the vestibule, inside the vestibule there are two structures, utricle and saccule. Okay, so these are two structures in the vestibule. So vestibule we can actually divide into two parts: the outer bony part, outer bony, okay, a part or outer bony labyrinth. Makes sense? And the outer bony labyrinth is filled with a fluid that is called the perilymph. Okay, and the inner part has the utricle and saccule. Okay, these are the membranous structures, and the utricle and saccules are filled with endolymph. Okay, which is rich with potassium, high potassium concentration and low sodium concentration. Okay, but perilymph has high sodium, low potassium. Okay, this is important. Okay, now we'll see these two membranous structures in the vestibule, and inside these two structures we have the sensory epithelium. Okay, and in the sensory epithelium you have the sensors or receptors. Actually those receptors are called the vestibular hair cells. Inside the cochlea, which is responsible for sound, you have the cochlear hair cells. Those are the receptors. Inside the utricle and saccule, you have the vestibular receptor cells. Those are also called vestibular hair cells. So all those are hair cells. Okay, so now we'll see how the hair cells, vestibular hair cells, are activated. Okay, if you see inside the utricle, what you will see is the sensory epithelium is located in the floor. So let me just draw. If this is, this box is showing the utricle, okay, this is the floor, right, and on the floor you have the receptors or hair cells, okay, here, and the fluid is inside endolymph. So when you move your head in horizontal plane, sideway or forward, backward, okay, or tilt your head, what happens? The fluid moves like this, right? Either this way or that way, but horizontally, or sideways, or forward and backward, but on the transverse plane. So what will happen if the fluid moves this way? The hair cells on the floor of the utricle, since the hair cells or receptor cells or sensors are on the floor, the fluid movement will activate the hair cells, okay, and the signal action potential will be created. Okay, so same way the neurotransmitters will be released and action potential will be taken to the vestibular nucleus. Okay, so that is the utricle. Now if you see inside the saccule, the location of the hair cells or sensors or receptors, whatever you say, is not in the floor but here, the posterior surface, and they are facing forward. So you have the receptors, hair cells here, and they are facing forward. So now you just think that if you move up and down vertical, okay, then what will happen? These, since the hairs are sticking out forward or outwards, what will happen? These hair will bend, and that will activate the hair cells in the saccule. So this is saccule, this is utricle. Makes sense? So utricle hair cells will be activated by horizontal plane movement, and also head tilt, and the sacule hair cells in the sacule will be activated by vertical movement, okay, and will be taken to the vestibular nucleus. So these two structures are, taking signal uh, or being activated by two different types of linear acceleration. This is horizontal and this is vertical, okay, up down. So we can say, like, when you are driving, if you suddenly brake, push the brake, then your head moves forward, right? So that will activate the utricle. And if you are um, going up in an elevator, and suddenly if the elevator stops, then you tend to move down. Head tends to shake or moves down, and that up or down movement will activate the sacule, okay. Or if after the lift stops, if it starts again downwards, then your head or body moves a little bit upwards, and that will activate the sacule, okay. So now a little more about inside the utricle and sacule. So what I'll do, I'll see the location of the epithelium, sensory epithelium inside the utricle and sacule, okay. The structure, let me take the dark color. So this structure where you have the sensory hair cells is called the macula. And if you see the structure of a macula, this is the basement membrane, connective tissue, supports the sensory epithelium, okay. And on the basement membrane you have the supporting cells. So these are the supporting cells to support the hair cells, okay, supporting cells, okay. And on the supporting cells you have the hair cells, okay. So these are the hair cells, and you have the stereocilia like cochlear hair cell, same way. And the tallest stereocilia is the kinocilia, you already know that. So this is the kinocilia, okay. So these are stereocilia, okay. And these are the hair cells, vestibular hair cells. And also there is another type of cells, those are also important. These cells are called the vestibular dark cells. So these are hair cells, vestibular hair cells or receptor cells, and these are the vestibular dark cells, okay. These vestibular dark cells secrete potassium. So that's why in the endolymph, right, you have the endolymph here inside, the potassium is high, okay. Potassium is high, okay. And the structure is very similar to the cochlea. There is a membrane here, rests on the hair cells, and that membrane is called autolithic membrane. So this is the soft gelatinous membrane called the autolithic membrane. Okay, and the hair are inserted or attached to the autolithic membrane, and this autolithic membrane, which is gelatinous, is actually formed by mucopolysaccharide. Okay, mucopolysaccharide. And inside the autolithic membrane you have some important chemical crystals. Those are called autoconia. So let me draw those crystals. So inside the autolithic membrane you have the crystals of calcium carbonate. Okay, and these are called autoconia. These are what? Calcium carbonate crystals. Calcium carbonate crystals. Okay, and it gives more weight to the autolithic membrane. So when it will move due to acceleration, it will not be displaced in other directions. So it gives the motion, but it's like the motion is controlled motion. Makes sense? Okay, so when you so this is the structure of that sensory epithelium inside the utricle or saccule. Now when you move your head in horizontal plane, then these hair cells inside the utricle will be activated. How? The fluid will move the stereocilia, also will move the membrane, autolithic membrane, and the hair will bend. And you know that coniocilia is the tallest one, and these are all connected by tip links, right? So this is kinocilia, and these are stereocilia, and they are connected by tip links. I explained that. And when the cilia will move, the tip link will open the channels. So the potassium will get in, because in the endolymph the potassium concentration is high, and when the potassium will get into the hair cells, then the signal action potential will be generated by the release of glutamate, and that will produce the action potential, right, in these afferent fibers. So a lot of action potentials will be generated and will be taken to the vestibular ganglion. Okay, so this is the vestibular ganglion, like cochlear spiral ganglion. You have the cells, and they will take the signal and then will send outwards to the vestibular nucleus in the brain. Okay, so that is what happens inside the utricle. Inside the saccule, same way, only these hair cells will be activated by the vertical acceleration. Okay, vertical movement up or down. Okay, now one thing is very interesting and important is I am showing in one side's utricle, right? But you have two ears. So in the other side, what will happen? The hair will move to opposite direction. Makes sense? Because when your left ear moves laterally, that means right ear is moving medially, so opposite direction. So inside the other utricle, the hair cells will be hyperpolarized, that means deactivated. Okay, so these hair cells will be heavily deactivated. So very little or no, action potential will be generated here. Okay, no action potential. And in this side, lot of action potential. So signal will be taken to the brain from both ears, right, at the same time, because you have two ears and both are moving in horizontal acceleration. So now you see this one is bringing a lot of action potentials and this one is no action potential, and your brain structures can subtract these two and get the signal from the difference between these two. In this case the difference is very high, right? Now if you move your head to the opposite direction, then these fibers will be activated and a lot of action potentials in this side, but in this side no action potential. Okay, so your brain can detect that by counting the difference of the action potential. Make sense? So that's how the vestibule works to detect the acceleration, linear acceleration in horizontal and vertical planes. That's why you have those two structures. Okay. Now I'll talk about the semicircular canals. There are three semicircular canals. One is like towards your outer ear laterally, right? That is lateral or horizontal canal. Another is towards backwards, towards the back of your head, and that is the posterior or inferior semicircular canal, and another is anterior or superior. It's like upwards. So these are three semicircular canals. Now the question is why you have three semicircular canals, right? It is very important and very interesting that you have three semicircular canals, and these three semicircular canals are positioned in a way that it covers the rotational acceleration or movement in all different directions. Okay, so all different rotational accelerations are covered by these three semicircular canals. Okay, and they are positioned that way, and each one is separated by 90 degree if you just measure, and that's how it covers all different angular or rotational accelerations. Okay, now if you see the beginning of the semicircular canals, they start from the vestibule, and that area is a little bit wider or dilated. That is called the ampulla. Okay, so the first part is slightly wider or dilated, and that is the ampulla of the semicircular canal. Okay, so these are ampulla. Okay, and the semicircular canals are picking up the signal of rotational, not linear, acceleration. Okay, so when you rotate your head, the sensors inside our hair cells, inside the semicircular canals will be activated and will be able to detect the angle of rotation. Make sense? So now I will show you the structure inside structures of a semicircular canal. Okay, So if I just take this part and see, this is the ampulla, okay, and this is the canal, okay. So ampulla, canal, and inside the canal you have the duct, semicircular duct filled with endolymph, okay. Same fluid we talked about, this fluid in the cochlea and vestibule, endolymph, right, rich with potassium, okay. And the outer part again is bony, and inner part is the membranous part, okay. So bony labyrinth is the outer part and membranous labyrinth is the inner part. Bony labyrinth, okay. And inside the bony labyrinth you have the membranous labyrinth. And if you see the structure of a membranous labyrinth, you have the basement membrane and the hair cells, right? So basically the structures in the sensory epithelium, same structures are there, right? These are the hair cells, but these hair cells will be activated by rotational motion or movement or acceleration, okay. So also you have supporting cells around the hair cells. I'm not drawing those. So hair cells, okay. And if you see on the top of the hair cells you have again a mucopolysaccharide structure. It is a huge mucopolysaccharide soft gelatinous structure. In this case this structure is called cupula. Cupula, okay. Now when you move your head in, you rotate your head like sideways, right and left, what happens, the endolymph from here will rush into the ampulla, make sense, and that will move the hair or bend the hair, and if the fluid moves this way, the hair will open the ion channels because the kinocilia will move that way, right, and that will open the channels by moving the caps that covers the channels, so tip links will be stretched and that will open, same way, same mechanism. However, this is important, in the other side you have the same semicircular canal. For example, this one is the lateral one, this is also lateral one in the other side, right? So when you rotate your head, the hairs of these hair cells will be activated, these will be inhibited here, same way we discussed in the vestibule, right? So lot of action potential here, but no action potential due to hyperpolarization here, okay, no. So the difference will be big, makes sense, and that your brain can detect or calculate that difference, okay. So the in this case, for example, the endolymph will move, rush inwards. In this case, outwards, opposite. So the here, deflection of here or bending of here will be opposite. So these will be activated, these will be inhibited. Okay, so that's how the brain will determine the angle or direction of rotation. Makes sense? So those are a few things you need to know about the cochlea, vestibule, and the semicircular canals. The last thing that I didn't explain is that if you see inside the cochlea, you have the basement membrane, right? And if you see the basement membrane, the hair cells at different locations on the basement membrane are activated by different frequencies. So this is the basement membrane, and basement membrane has attached side or fixed side or end and free end, which is the apex. Okay, is free. So just remember, the hair cells located near the fixed side or attached side on the basement membrane, these hair cells will be activated by high frequency sound. Okay, and those hair cells are sitting on the free side. These will be activated by low frequency sound. Okay, and the middle ones will be activated by intermediate frequency or medium frequency sound. Okay, so that's how the hair cells on the basilar basement membrane or basilar membrane, this is the basilar membrane, are activated by high frequency to low frequency. Okay, from the attached side or fixed side to the free side. Okay, it is like, piano. Those who know about piano, you have high note, medium, and low notes, right? So high notes are here, medium here, and low frequency notes are here. Okay, so when these cells will be activated, you will listen high frequency sound. When these receptors or hair cells will be activated, low frequency sound. Okay, the cochlea is responsible for sound. So if any damage occurs in the cochlea or death of the cochlear hair cells, that will do what? Will cause deafness, and that is called the sensory hearing loss. Why it is called sensory? Also called sensory neural, because the hair cells are the sensory receptor cells, and those are destroyed or die. Okay, and once the hair cells are destroyed, so far we know that no new hair cells are generated. Very recent studies have indicated that few hair cells could be generated, but we know that there are no stem cells in this area. So that's why the person needs cochlear implant to improve the hearing. Okay. Another type of hearing loss, which is just due to blockage, like any wax is formed inside the outer ear canal that blocks the sound waves, or if any fluid accumulates in your ear or infection. So the sound waves will not be taken to the inner ear, to the cochlea, or a tumor. Those are the deafness called the conductive hearing loss. So there are two types. Conductive hearing loss is due to the blockage in the ear, and if you remove the blockage, that person will be able to hear fine because his hair cells are still alive and active, right? But in sensory neural hearing loss or sensory hearing loss, since the hair cells die or are destroyed, it is difficult to fix. Okay, so I'll stop here, everybody.